When moss is boss in fixing crime, there is a popular belief among criminal investigators that a criminal always leaves clues behind. Scientific research now suggests that offenders may also carry those clues with them, sometimes in unexpected forms. One such form is moss, a small non vascular plant that grows in dense mats across a wide range of environments. Moss is increasingly being examined by researchers as a supplementary forensic tool capable of linking people and locations to trace botanical evidence. In 2009, employees of the Burr Oaks Cemetery in Chicago, Illinois, were accused of exhuming buried remains and reselling grave plots. Moss samples recovered alongside discarded human remains were examined by researchers at the Field Museum in Chicago and identified as Physidens taxifolius. By analyzing the moss fragments, the investigators determined that they have been buried for less than 24 months. This finding contradicted the suspects' claims and helped establish their presence at the crime scene. While the case did not involve homicide or missing persons, it demonstrated how Moss evidence could provide independent information that supports or challenges investigative timelines. Interest in such applications has been renewed following a large scientific review published in Forensic Sciences Research 2025, Volume 10, Issue 3. The study, titled Green Clues Unveiling the Role of Birophytes in Forensic Science, was conducted by 15 researchers from 11 universities and agencies in the United States. It examines how birophytes, particularly mosses, may assist for forensic investigations, while stressing that their use remains largely experimental. Biophytes and forensics. Biophytes are small, non flowering plants that lack a vascular system and include mosses, liverworts, and hornworts. Although these groups share several features, the study notes that nearly all forensic research to date has focused on mosses alone, which account for about 12,000 to 13,000 species globally. Researchers point out that moss fragments are easily detached and can adhere to shoes, clothing, tools, and vehicles. Once transferred, they may persist in soil, dust, or debris collected as evidence. This makes moss potentially useful in establishing associations between individuals too. Between individuals and specific locations, particularly when combined with other forensic findings. So, in experimental evidence, several experimental studies support this possibility. In one controlled experiment, researchers examined how well plant material adhered to footwear. After participants spent 24 hours walking through forested terrain, followed by 4 hours on gravel or paved surfaces, moss fragments were still present on 28% of the shoes. A total of 22 moss shoots from 5 species were recovered, indicating that such material can survive prolonged movement. Another study explored whether moss DNA remains suitable for analysis after long term exposure. Fresh samples were stored in paper bags in an outdoor shed in southern Finland for 18 months, during which temperatures ranged from minus 28 degrees Celsius to 25 degrees Celsius. Genetic material extracted from these samples yielded reliable genotyping results, suggesting that DNA based identification of moss fragments may be feasible even after extended environmental exposure. The study also notes that mosses display a high tolerance to environmental stress. While they are commonly associated with damp conditions, many species can survive periods of drying and resume growth when moisture returns. This resilience increases the likelihood that fragments recovered from evidence remain identifiable if collected and preserved correctly.